Okay. Thank you, DJ, for the introduction, and thanks to the Fudian community for giving me this opportunity to present my research work. So today, my research project has been still in this technique development phase. So I would like to talk specifically about the new DNA fish method that we've been developing. Especially I want to show you in the first part of my talk, how we can achieve high resolution DNA genomic imaging with this tool. And there's a second part of my talk, which you will learn from this presentation. So this talk will be more technical oriented. I have a lot of preliminary data, but hopefully this will be interesting to this community. Okay. So over the past decade of this tremendous amount of efforts this Fudian community have put in this past decades, we have already known a lot about the genome organizations in nuclei, especially genome organized in this hierarchical manner. We are specifically interested in the length scale at chromatin loops and domains, because there are a few interesting biological processes that happens at this scale. For example, DNA repair has been a major topic of our research in the HA lab. It has been shown that it creates this loop structure at this megabase scale, and of course enhancer promoter interaction, gene activation. This has been like a very active topic for this community. Happens at the scale as well. It also has been shown that the VDJ recombination in the B cell happens at this scale. So if we look into the tools that people have been using to study the problems at this scale, high-C conformation capture is definitely one of the go-to tool. But in our lab, we want to also look into the chromatin, the imaging side to complement this information. So on the imaging side, pioneer by a group of people in Xiaowei's lab, Alistair, Stephen Wang, Long Kai, they have come up with this similar method. The idea is that we look at a region of our interest, a piece of genome sequence. We divided them into small segments, and we image one segment at a time by sequential round of imaging. We can afterwards reconstruct the 3D conformation of this DNA structure. I think one benefit of applying this method is that it's easier to achieve this multi-model information. So for example, in Alistair's one of their work, they showed that transcription activity correlates with the genome conformation. So we can do RNA, DNA imaging at the same time. And in this picture, actually, the next level of the technique that we wanted to add on is to ask, can we actually include the information of a protein into this picture? Because we know that the protein, for example, transcription factors regulate transcription output, and also SMC proteins or CTCF boundary elements that actually they regulate the genome structure. Right? So it would be really good that if we can include the protein information into this imaging scheme as well. So there are some recent progress where people try to include the imaging, the protein side into this multi-model imaging. However, the imaging methods that included in this protein imaging has been shown to be limited to mostly large structures of inside the nucleus, because it's actually really hard to measure regulatory proteins such as transcription factor, SMC protein, DNA repair protein, for example, alongside with DNA fish. The major reason is that traditional DNA fish method requires heat denaturation to break up double-strand DNA for the DNA fish probe to hybridize, and apparently this heat treatment make it pretty difficult for protein imaging because it can disrupt the protein structure. So here I'm just trying to highlight like a few limitations of traditional DNA fish methods. One, as I already mentioned, being the heat treatment, heat denaturation. It prevents the quantitative protein imaging. And also people have been shown that heating the cell above 90 degrees Celsius has a potential to disrupt the the fine structure of the nucleome. Another thing about heat denaturation is that it's global, it's non-specific, so it actually opens up the entire genome. So in this case, you increase the non-selective binding of the DNA fish probe, which contribute to the background signal, preventing like really high resolution genome imaging. Another issue of this traditional DNA fish method is that when we denature the double strand, the non-target strand might be still around our target strand. So the non-target strand can potentially compete with the probe binding, which might lower down the probe hybridization efficiency. So some of these questions, this is a known problem of the traditional DNA method in the field. Some people have already tried to come up with some methods to address part of these problems. So one work from Robert Sinner's lab, where they use Cas9 decase fusion with fluorescent dye, they successfully demonstrate that Cas9 can be used as a genome imaging tool for repetitive sequences. However, in the same paper, they also show that if we want to look at the non-repetitive sequence, the signal to the background ratio is actually not really great. So this is where this previous graduate student, Yangbo from the Ha lab, is now a postdoc at Stanford, came in. So he had this idea where if we can use a Cas9 variant, it's a mutant Cas9, where it only cuts one strand of DNA. If we incubate this Cas9 with our fixed cells, it targets our specific genomic region. It actually exposes the 3' overhang, and this 3' overhang can be accessed to helicase. So if we then incubate the sample with the helicase can unwind the single-strand DNA to create this single-strand region for DNA fish probe hybridization. And this is the concept called goldfish, as TJ mentioned in the beginning. It's genome oligoplan via local denaturation. And this concept actually works pretty well in practice. You can see Yangbo demonstrate that this goldfish has much higher signal to the background ratio compared to catfish. It's almost like an order of magnitude improvement. So these goldfish methods that Yangbo came up with actually solve two problems of traditional DNA fish methods. One is that the helicase unwinding the DNA is it doesn't require any heat denaturation anymore. So we don't need to heat the cell up to above 97 degrees Celsius. Another thing is the Cas9 has a pretty high sequence specificity. So this entire goldfish scheme actually performs local denature. We don't open up the entire genome for probe hybridization. However, there's one question remains. This helicase actually, when it unwinds the DNA, it still leaves another non-target strand there. So this non-target strand can potentially compete with the probe binding. It's the same problem. There's another problem. So this original goldfish method actually creates a new problem. It only works with the methanol-based fixation, for example, methanol and acetic acid. And acetic acid, when acetic acid is used, it actually denatures most of the protein. So this method is still not so compatible with immunofluorescence imaging. So our goal is trying to see if we can solve these two problems in our next generation of DNA fish method. And when I look into this problem, I was actually quite inspired by two previous work which utilized the exon nucleus as a mechanism to generate single-strand DNA for probe hybridization. And so I asked if we can actually try to use exon nucleus to create single-strand DNA. Essentially, can we try to replace the helicase in this goldfish protocol? So this is the idea. So we still use Cas9 in this case, which cuts only single strand. And this single strand DNA can be exposed to exon nucleus, which digests to create a single strand region for probe hybridization. And because this new goldfish concept utilizes exon nucleus, we actually name it as goldfish X. And it actually works pretty well, as you can see. Of course, after so much try and errors and many iterations of optimization of our protocol. After we incubate the Cas9 nickase and switch to the exonucleus digestion, we can faithfully generate this single strand region for prop hybridization. As a control, if we don't use Cas9 but keep the entire protocol the same, we don't see any DNA fish spots. So these are actually pretty good, exciting results. So now the exonucleus actually solved the problem, one of the problem that the original goldfish left, which is it actually really generates single strand DNA templates for our prop hybridization. So now we want to ask, right, how's the performance of the goldfish eggs? So at the largest scale, largest segment imaging or labeling, for example, 7KB region, we actually found that the exonucleus performs pretty similar to the results from the HELIX case. And we can quantify the signal a bit. You can see because this is U2O cells, it's a polyploid. So a lot of time we found more than 4 spot for this specific gene beta-actin. But the point is, we can see the signal is actually very well separated from the background. Given this amount of signal, we are actually thinking perhaps we can try to use less amount of probe and we can still see the DNA fish spots. But by using less amount of probes, we can actually reduce the length of the segment that we actually label, and which allows us to achieve higher genomic labeling resolution. And so we started to test. I think now the current state of our genomic resolution for DNA fish is roughly like 2 kb. And we've been showing that the 2 kb labeling in our hands is actually pretty reliable. We can further shrink the length of the DNA segment that we label down to roughly 1 kb, 1.2 kb. We can still reliably see the DNA fish spots from these samples. We want to actually test the limit of this protocol. So we really actually just label very small segments. And now we saw that if we push the labeling down to roughly 300 base pairs, 350 base pairs with only 12 oligos, we can still see some spots in some of the cells. At the cost of the overall label efficiency, we can kind of lose some signal from some of the cells. But for the cells that works, I think it kind of recovers pretty well. The spots is actually pretty detectable. This is just another example showing the images of 300 base pair labeling, and you can see some of the spot here is actually pretty dim, but if I just on the right side, I'm showing you, the Z stack rolling over different Z planes, and you can see at the single plane level, it's actually pretty bright. It's pretty visible by eyes. This can be definitely captured by the computer program. So at this moment, we are actually quite confident that actually this method can achieve very high file resolution. This is another example. Before I was showing the data from labeling of beta-actin. There's another gene, MinSyn4. We can see when reduced from 2.3 kb labeling to 650 base pairs, we can still see quite bright spots. Roughly 330 base pairs, we can still detect the DNA fish signal. So I think at this moment we are convinced. I think the protocol can still we're still trying to further optimize the protocol. So it's still an ongoing effort. But at this moment, we're already pretty excited about this new DNA fish method, Goldfish X. It actually performs pretty well. So this concludes the first part of my talk, that we have come up with a new DNA fish method that can achieve high resolution. The second part of my talk, I want to address the second question, the second problem here. Because as I mentioned, because the use of the acetic acid denatures the protein, so preventing the immunofluorescence imaging. So can we simply replace the methanol-based fixation with formaldehyde fixation, because formaldehyde fixation is mostly used in immunofluorescence imaging. So maybe there's a chance that that these two protocols can be compatible with each other. And in our first try, I think this goldfish protocol actually doesn't really quite work with the PFA fixation, PFA fixed cells. But by playing around with this fixation conditions, we actually started to see some hope. I think there are under certain conditions, we can actually see a little bit DNA fish spots. And after like a series of effort into optimize this fixation protocol, at the end we actually recover most of the DNA fish signal in the PFA fixed cell. So we are actually very excited about this new progress. And here, this is another sample. We use DNA fish to target another gene using 2.3 kb with 83 probes. You can see the signal to the background is really, really high. We quantify the intensity is pretty well separate from the background. And this gene is much well behaved. So I think, if you detect, I think most of the cell we can see 4 spots. Some of them we can see 2. So yeah, this is a pretty good label efficiency. So now I think I've shown that this new DNA fish protocol can actually work with PFA fixed cells, right? So our ultimate goal is see if we can finally get the DNA fish to work with immunofluorescence imaging. And the answer is actually yes. So we have tried a few settings where in one case on the left, we label the larger structure within the nuclei, the nucleolus, and you can see the nucleus and DNA fish signal can coexist in one set of experiments. And these genes, they are actually, we're not expecting any colocalization or anything. We just want to see if these two protocols can actually work together. And in another case, we try to label DNA repair protein Ref51, and you can see, again, we can see clear Ref51 PANTA alongside with the DNA fish signal. So this shows that our protocol works pretty well with immunofluorescence imaging. More recently, we are working with our collaborators to see if we can label transcription factor and its genome target at the same time. And this data is actually really, really fresh. We got it 2 days ago, and we were really, really excited about it because you can see from on the left side, there are a few genomic loci, and we can see the transcription factor forms PANTA and actually colocalized with the target loci. So here we can see more examples, and this specific transcription factor is actually from an autoimmune system. It actually interact with multiple targets, so we only label one of the targets. So we can see the targets that we label, we can see enrichment of the transcription factor, but there are also more PANTA, but presumably they are interacting with other different targets. So now we have shown that this new Goldfish X protocol has the capability to label transcription factor and its target at the same time. So the next steps. So as I mentioned in the beginning, our lab has been pretty interested in study the mechanism of DNA repair. So we want to combine this new protocol with the apto control CRISPR tools that they also developed in the lab previously called very fast CRISPR. So idea is they want to generate the DNA double-strand break with optical control, which can synchronize a monopopulation of cell, and then we fix the cell at different time point and perform DNA fish imaging and protein imaging at the same time. And we can also, another thing that we wanted to do is trying to extend this DNA fish protocol to the sequential imaging so that we can map out the fine structure of chromatin around the DNA break site. In fact, we already have some preliminary results. In this set of data, we actually try to perform this apto-triggered, apto-controlled Cas9 cleavage. And then we fix after 3 hours of the UV activation. So on when we use goldfish to label the cut site, and then we also at the same time label gamma H2AX, which is the DNA break marker, and red 51, which is the DNA repair protein. So if we zoom into this data, we can see at the cut site, it colocalized with the DNA the DNA break marker gamma H2AX, and you can also see the red 51 accumulates around the cut site, so it shows the DNA repair was actually repair machinery was actually recruited at the cut site after 3 hours of the introduction of the break. And there's a 3-color colocalization here. You can see there are more examples in different cells. So this is a conclusion. Here we actually developed a new DNA fish method we term Goldfish X, and it has the capacity to achieve hundreds of base pair genomic resolution with decent label efficiency, and it's also compatible with protein imaging. So I guess this could potentially open up a lot of opportunity with this tool. So in the future, we want to combine this with a very fast CRISPR to study DNA repair mechanism, and we also want to extend this tool to sequential imaging to map out the fine structure of chromatin. Okay, so at the end, I would like to thank you for your attention, and I'd like to thank my mentor PI, TJ, and also the graduate students and postdocs that I work with from the lab. And this method actually has been catching a lot of interest from our collaborators, so they wanted to try to see if we can label the protein and DNA at the same time. So also thanks the input from our collaborator and the funding source.